Okay, hi guys. So yesterday I was talking about the digestive system and how large molecules are broken down and they're absorbed in the digestive system. We touched on a few times about these molecules, enzymes, which are involved in digestion. So we're going to talk in this video, to finish that section off, about what an enzyme is, how it works, and we'll touch upon why it's important. Now, on AQA, we'll learn about enzymes in the context of digestion, but really enzymes are central to any reaction that takes place in living things. So if you are doing a different exam board, we kind of might learn about enzymes in different contexts, but fundamentally they're the same things, they're doing the same jobs, it's just different reactions. So what is an enzyme? An enzyme is a biological catalyst. And what that means is a catalyst is something that speeds up a reaction without being used up in the reaction itself. And biological means it's a living thing. So enzymes are proteins. They are made up of amino acids. Talked about that in the video I made a couple of days ago. And they have a specific tertiary structure. They have a specific 3D shape. And that 3D shape gives them a unique area called an active site. Now, that active site is complementary or specific to the shape of a particular reactant. And when we're talking about reactions with enzymes, we more often use the term substrate than we use reactant. So if this is the enzyme here, the active site at the front, this is the substrate or the reactant. And you can see the substrate fits perfectly into the shape of the active site. Different substrates or different reactants that the enzyme isn't specific to would not be able to fit into the active site and would therefore not be worked upon, not be catalyzed by the enzyme. So what we get then as a result of the reaction are the products. And the products are the thing, or the material, the substance, chemical that is produced in any reaction. So the substrate goes in, the products come out. And you can see the substrate looks like this and the products broken down into smaller chunks. So the whole process there that's being described, we often refer to as the lock and key theory. And what that means is the enzyme is acting as the lock and the key is the substrate. The substrate fits perfectly into the lock. And if you had the wrong key going into a lock, it wouldn't turn, it wouldn't fit into the lock. So that lock and key theory describes that process. Now, what's going on there is that the enzyme really is lowering the amount of energy that's needed for a reaction to take place. We call this the activation energy. And we learn a little bit more about this in chemistry and kinetics of a reaction. And this kind of goes across different exam boards, and understanding this is more of a requirement on certain exam boards than others. And essentially, activation energy is just the amount of kind of clout that the molecules need when they come together in order for the bonds to break and rearrange to give us the new product. If we're going from this, as I drew above there, here and here, to form these new products, then what's gone on here is that bonds have been broken, new bonds have been made and rearranged. So when products are formed from reactants come together, there is a level of energy that's required in order for that to happen. And if they don't reach that amount of energy, then the reaction can't take place. And what an enzyme does is it lowers the amount of energy that's needed for that. We might kind of recall from chemistry a graph that looks like this. You'd see delta H energy up the side, and we have an energy profile of what goes on within the stages of the reaction. Here we have the reactants at the start. Here we have the products. And instead of the line just going down like that and across, which is what you'd expect, what we'd actually get here is this increase in energy until we reach this point here. That increase in energy is signifying the fact that these particles need energy to get things going, to break the bonds, to make new bonds. And when we reach that point, then we have enough energy for the reaction to take place. That activation energy, if we use an enzyme, is lowered. So we don't need as much energy in order to trigger a reaction, to cause a reaction to take place. So going back to the original definition of what we said an enzyme was, a biological catalyst, something that speeds up a reaction. How does it speed up a reaction? Well, what it does, it means that we need less energy for reactions to take place. Therefore, more reactions happen with less amount, so reactions take place faster. So all of those key terms have got to be used comfortably in interpreting graphs, interpreting data, but quite often these are long answer questions, guys. So 5-6 marks, depending on which paper, which board you're doing, and describing how reactions take place. So you should practice a few kind of longer answer questions and describe how an enzyme works and how it breaks down products and in what situations it's used for. There are a few specific examples to digestion we need to know as I'm going through the AQA spec here. We talked about different food molecules last lesson in the video on the digestive system. We've got to know a few specific type of digestive enzymes, carbohydrases, proteases, and lipases. These are all released in the pancreas. So for example, carbohydrates like amylase breaks down starch, breaks down carbohydrates. So I'll just put carbs. Proteases break down proteins into amino acids, and lipases break down lipids into fatty acids and glycerol. And that was discussed in the previous video. And we should know where these are made and how these are examples of enzymes. And what they are doing is they are breaking these longer molecules down into smaller molecules, and those smaller molecules. Okay, the next important feature of enzymes that we've got to mention that is across spec boards is denaturing. And what denaturing means is that under extreme conditions, namely heat or extreme pH values. So if you're not happy with what pH means, it means the level of acidity or alkalinity of a solution. So remember, the pH scale goes from 1 to 14, 7 being neutral. So in really extreme pHs, i.e. really low or really high, the enzyme will denature. But what does denature mean? Well, I said earlier that an enzyme is a protein, and a protein is made up of a chain of amino acids all joined together. And when a chain of amino acids come together to give us this 3D shape of the enzyme, it has this specific structure. Now, if the heat gets too high, or the pH gets too low, too high, too extreme, then what starts to happen is that some of these bonds start breaking apart as the result of those conditions. And if they break apart, the structure, the overall structure of the enzyme is going to change. And that's going to give us, ultimately, with the enzyme, a different active site. Now, if the active site suddenly changed shape, as a result of the change in tertiary structure or 3D shape, the substrate that would have fit into that enzyme and therefore being able to be broken down or put together, that reaction can no longer take place because it no longer fits into the active site. So when an enzyme becomes denatured, it is permanently damaged. It's not used anymore because the active site is no longer any good. It can't do what it's supposed to. It can't lower the activation energy. And we can draw this on a graph. So if we look at the rate of reaction as we increase the temperature, so let's say this is kind of going up by 10 degrees, roughly speaking, 40, 50, then 60. And this is the rate of reaction. So that's perhaps the amount of product that's being produced, or the amount of substrate that's being used up. But let's just call it rate for now, how fast that's taking place. So often these are describe questions on GCSE exam papers. Describe what's happening in the graph, explain what's going on in the graph. And if we're looking at the increase in temperature, what we generally see would be this shape here. And what we're seeing there is that as the temperature increases, the molecules that are coming together to react, the reactants, have more and more kinetic energy. They're moving faster. So therefore, the number of successful collisions and the rates of reaction are increasing. And that continues to happen if we're looking at something in the body until about body temperature, which is about 37 degrees. And we reach a point here, which we call the optimum temperature, the temperature at which the reaction is taking place at its optimum rate as fast as it possibly can. What will happen if the temperature continues to increase past that point, I'll draw this in a different colour, is the rate of reaction will start to fall. And that's because this has happened. The enzymes now are starting to look like this. They're starting to break down. And that causes less successful collisions, less reactions, because there are fewer enzymes that are available to trigger that to happen, to lower the activation energy. So at this point on the graph, what has happened is that the enzymes have denatured, the rate of reaction lowers. Now, if I take this off and we look instead, not at temperature, but we look at pH, and what we'll do is we'll look at an enzyme that you might find in the saliva, amylase. So amylase is an enzyme that breaks down starch. It's a very common one to see on exams, and we're going to talk about the required practical on AQA, practical 5, which is looking at the effect of this change in pH on the rate of reaction of amylase. So amylase in the saliva will be about neutral. Your saliva is neutral, about pH 7. So that's going to be its optimum temperature, optimum pH, sorry. So the optimum pH, the optimum rate of reaction is going to be up here for saliva. Now if we, for salivary amylase, sorry. If we lower the pH, then the rate of reaction is going to drop. And equally, if we increase the pH, the rate of reaction is going to drop. And that's because this process, this denaturing, is taking place because that change in pH is changing the charge of the amino acids, the bonding that goes on in there, and that in turn breaks the bonds and therefore changes the 3D shape, so we get denaturing taking place. Now if we looked at a different enzyme, one that's in the stomach, pepsin, looked at that in a previous video, that might be closer to pH 3. So the optimum is going to be up here, and then if we decrease that even further or we increase it, the rate of reaction is going to fall. We're moving away from the optimum. This process is beginning to happen, denaturing. We looked at an enzyme that might be in the small intestine, protease or carbohydrases, we talked about earlier. In the small intestine, we get very alkaline conditions to neutralize the stomach acid that's coming through, the hydrochloric acid. So what we'd see would be a graph that would be more like this, perhaps closer to 11 or 12, something like that. So the optimum pH is going to be higher, and if we go above or below that, this process takes place. So there is a required practical on AQA where we look at the effect of changing pH on amylase. Now, the reaction here is the one that we see below. Amylase breaks down starch into maltose, which is a type of sugar, a simple sugar, and that's all we need to know at GCSE. So the starch is the substrate, the sugar, the maltose, is the product. And if you remember from the video on the digestive system, the previous required practical 4, asks us to understand how we test for these. So we use iodine to test for starch. That goes blue or black. And if there is no starch present, the iodine will remain orange, which is the colour it begins at. So what we do in this practical is we take a spotting tile, which is basically just a plastic dish. It's got a number of kind of little wells in it, and you can drop small amounts of liquid in to test certain chemical reactions. And what we're going to do is we're going to take a beaker of starch here, and we're going to add in some amylase. And then the independent variable here, the thing that we're testing, is pH. So what we're going to do is we're going to use something called a buffer. A buffer is just a salt solution that maintains a pH at a constant level. So we might try a few different pH buffers. pH 4, which is marginally acidic, pH 7, pH 10, something like that. Depends on what's available in school and how many different kind of ways you've got to set it up. So let's say we've got a buffer of 4 here. Actually, let's change that. Let's say we've got a buffer of 7, which is about the optimum for salivary amylase. So if we take a pH 7 buffer, that's going to keep the pH constant. We've got the starch in the amylase, and this reaction here is beginning to happen inside the beaker. So we're starting to get the starch broken down, but it's going to take a little bit of time to get going. So what we're going to see to begin with is that the starch is present. It's not being broken down. So initially into each of these wells, we are going to put a tiny drop of iodine. Going to add a little bit in to each well, like this. So it's all there ready, waiting. And then every 30 seconds, we're going to have a stop clock going. Every 30 seconds, we're going to drop in some iodine, pick up on some of this starch solution here with the amylase and the buffer, and see if there is any starch present. Now maybe initially, because it takes a little while for this to get going, the starch hasn't been broken down, so the iodine is changed to blue or black, so we get that colour change. And then maybe after 30 seconds that still happens, maybe even after 60 seconds, 90 seconds. But then what's going to start to happen is as we get further down, because we're at pH 7, which is about the optimum pH for amylase, it's not going to take that long for the action of the amylase to get going and take effect. So we're going to start to see a little bit of starch, and then perhaps, and then perhaps at that point, less and less of the starch is present because it has been broken down. And at the point where there's no starch present, or we get less of the starch, we record that time. We perhaps repeat the experiment a few times to make sure that it's a reliable result. And that enables us to record a time roughly for how fast the reaction took place at that pH. And then what we do is we reset the whole experiment. We start off with the tiles again, and we take out the colour. So let's take out that from inside. We're going to start off with the iodine in the dabber dish, just like we did before. Just colour that in here. We've got a little bit of iodine in each one. And then we're going to take a tiny drop of the starch solution. But this time the starch solution has been left in buffer 4, so pH 4. So that's different to pH 7. So now we're testing to see if the acidic pH has more of an effect. So every 30 seconds, we add in a little bit of the starch solution with the amylase. And what we should see is that it takes longer for the starch to start being broken down. So it perhaps takes, maybe I've got 240 there, every 30 seconds. So at that point, when the colour change no longer happens, that means that the starch is no longer present and has been broken down within the beaker. It has had enough time for it to be broken down. We repeat that a few times, we'll see if we get a result. Now, sometimes we're asked questions about the equipment that we'd use on this. Sometimes we're asked questions about, well, you and me might look at that dabber dish and say, well, I can see a little bit of dark blue colour there, and somebody else might not be able to see that dark blue colour. So there is an element of human error there also in looking at that. So quite often we're asked questions about reliability. And perhaps if we're only testing 3 pHs, like 4, 7, and 10, it might be that, and we're trying to find the optimum pH of a particular enzyme, it might be that 7.5 is better, or 7.9 is better. So by using quite large intervals in our experimental variable, our independent variable, that also presents a problem in the accuracy of your results. In all the experiments that we do, no matter what exam board you're looking at, these terms, reliability, accuracy, precision, are all things that you're going to be tested on. Okay, so that concludes the enzymes video. I'm going to go on now and have a look at the questions that you might use. So if I were studying AQA, I would go to the AQA GCSE section of Physics and Maths Tutor. If this is the first video you've looked at, this is a really fantastic site. It's got lots of useful resources, and I'm just using it basically as they have free access to question banks that you can use that are applicable. Rather than trying entire papers, you can try just specific topic questions that I'm going to show you here. So I'd maybe kind of plow through a few of these. Question 1 has a nice question here, experimental technique on rates of reaction in enzymes. So I'd maybe kind of have a look at that, and then I would use MS, the mark scheme, to check that I was on the right lines. Remember, please stick any comments in the videos that I've got on the YouTube channel, or I'm posting them on Facebook, on Instagram. If you stick a comment down, I'll try and get around to answering it and answer it as best I could. But perhaps you saw a question that you definitely were sure.